Hello to my family, welcome back to the channel. So, today we are going to discuss scientific methods in psychology. So, in our first sessions, we have already discussed that psychology is basically a scientific discipline. So, it is based on experiments and scientific studies. We are going to study the scientific methods that we use in psychology and what are the steps that we are going to follow in while doing experiments while doing different researches in the future. So, the scientific method involves a series of steps that are used to investigate a natural occurrence, right? So, anything that is occurring naturally has to be investigated deeply and thoroughly, but with the help of scientific methods. So, what are scientific methods? The experiments and different observations that we make on the basis of experiments, these are all the parts of scientific methods, right? So the scientific methods have different steps, which are problem or question, observation, research, formulate a hypothesis, experiment, collect and analyze results, conclusions, and communicate the results. So these are seven steps in scientific methods. So let's see one by one what is it. First one is problem question. So develop a question or problem that can be solved through experiment. Now, initially, before conducting an experiment or before going to start a research, there is a question in a researcher's mind and in a psychologist's mind that what is the most probable uh, observation that needs to be tested, and there is a need of doing experiment related to that observation. So this type of observation and thinking patterns generates a problem, right? So what is the basic problem? That is the identification of the basic problem. That is the first step in scientific method. Then comes observation research. Make observations and research your topics of interest. If a specific problem is highlighted, then we need to do some observations related to that problem. So, observations from the environment is the second step in the scientific methods. Third one is formulate a hypothesis. So, what is a hypothesis? Hypothesis is basically a tentative guess. It's going to be either it is going to be accepted or rejected after research, right? So predict a possible answer to the probable or to the problem or question. For example, if soil temperatures rise, then plants growth will increase. So here, if soil temperature rise, so this is the problem or this is the observation that needs to be tested. So, if soil temperature rises, then plant growth will increase. So, basically, plant growth is dependent on the temperature of soil. So, if soil temperature will increase, then plant growth will also increase. And if there is no increase in the soil temperature, then there is no growth in the plant. So, basically, it needs to be tested. So, for that, we need to do different experiments to test this. Either it is directly related to the soil temperature or not. Either plant growth is dependent on soil temperature or it is independent of that. So next one is experiment. So to find out the solutions for this hypothesis, to check this hypothesis, we need to do different experiments. So develop and follow up procedure, include a detailed material list. The outcomes must be measurable, quantifiable. So in experiment, whatever the experiment is, all the variables or the measurable elements should be quantifiable as well. You can count them. They can be calculated, right? So basically fourth step is to develop to design an experiment in which we can prove our hypothesis. Either it will be approved, accepted, or it will be rejected. So the fifth one is collect and analyze results, modify the procedures if needed, confirm the results by retesting, include tables, graphs, and photographs. So, after doing experiments, we have to give the results in different graphs, in the forms of graphs and tables and photographs, etc. So, we have to collect all the relevant results and analyze the results accordingly. So, after analyzation, the interpretation of result is a must. So, after interpretation, that either your hypothesis is going to be accepted or it will be rejected. Either there is a direct relationship of soil temperature with the plant growth. either it is not related, either plant growth is not dependent on temperature of soil, right? So the sixth step is conclusion. Include a statement that accepts or rejects the hypothesis. Make recommendations for further study and possible improvements to the procedure. Now, in conclusion, we have to give a single idea that either our hypothesis is accepted or it has been rejected, right? So, the seventh step is communicate the result. Be prepared to present the project to an audience. Accept questions from the audience. So, whatever the results might be, either they are in support of your thoughts or your hypothesis, or they are against, either it is accepted or it is rejected, your observation, but you have to communicate your results. Whatever the findings might be, you have to communicate the results to the audience as well. So, this is the seventh step in the scientific method. So, let's put our knowledge of the scientific method to a realistic example that includes some of the terms you'll be needing to use and understand. So, basically, we have discussed the seven steps that are the part of scientific method. Now we are going to discuss them again, but with the help of examples to clarify it in depth, right? So, in depth analysis is a must. So the first one is example is John watches his grandmother bake bread. He asks his grandmother what makes the bread rise. She explained that yeast releases a gas as it feeds on sugar. John wonders if the amount of sugar used in the recipe will affect the size of bread loaf. So basically, now there is a problem and there is an observation as well. John has already observed this thing that what is the thing that is required to rise the bread. So he asked his grandmother. She said that yeast releases a gas and it feeds on sugar. So basically, the amount of sugar used in the recipe will affect the size of the bread. So if we increase the amount of sugar, the loaf, the size will also increase, right? So the bread will also increase in its size, in its loaf. So this is the problem statement that sugar itself is independent. It is not depending on something, but on the other hand, the loaf, the bread size basically is depending on sugar. So he needs to check either increasing the amount of sugar will directly affect the bread loaf or it doesn't impact in any way, right? So this is the problem statement. This is the first step. So for that, he did different observations. He read articles. He read different researches. So John researches the areas of baking and fermentation and tries to come up with a way to test his question. He keeps all of his information on this topic in the journal. So whatever he was getting, whatever ideas he was getting from articles, by reviewing different books, so he was just trying to collect that information, and he was maintaining that information in a journal. So is formulating a hypothesis. So we have already discussed that what is a hypothesis. Hypothesis is a tentative statement. So after talking with his teacher and conducting further research, he comes up with a hypothesis. If more sugar is added, now this is his hypothesis that if more sugar is added, then the bread will rise higher. So basically, here is independent variable and dependent variable. What is independent? That is not depending on any other factor. So if more sugar is added, so if we see the variable that is sugar, that is not depending on anything. I mean, there is no factor that is impacting on sugar. We will increase. It's basically on us that if we want to decrease the amount or we want to increase the amount. It means that sugar is independent. It is not depending on some other factors, right? So if more sugar is added, then the bread will rise higher. But here the bread that is not independent, that is depending on sugar. If we increase the amount of sugar, the level of bread, the loaf of bread will be increasing, right? So we want to see that if is there any impact of increasing the amount of sugar on bread, or there is no impact. So this is the hypothesis, right? So the hypothesis an educated guess about the relationship between the independent and the dependent variable. As we have discussed earlier, that what is a dependent and dependent variable. These variables will be defined in the next few slides. That a independent variable or the independent or manipulated variable is a factor that's intentionally varied by the experimenter on x-axis. John is going to use 25 grams. Then in next experiment, he's going to use 50 grams, then 100 grams, then 250, then 500 of sugar in his experiment, right? So basically here he wants to check that is there any impact of adding the sugar on the size of the bread or not. So basically, what is the independent variable, which is a manipulated variable? Now, what is a manipulated variable? Which can be changed with respect to the thinking of the experimenter. If experimenter wants to change the quantity or anything related to the independent variable, he can change. So he can manipulate that variable. Like in the example we have mentioned, first initially basically he used 25 grams of sugar. So then he manipulate that quantity and then add 25 grams more. So it becomes 50 grams. Then he use 100 grams. So every time he is doing experiment, he is adding the quantity of sugar, right? So he is manipulating the quantity. So that is why independent variable is always a manipulating variable. And this is all about the independent variable. Any variable that is not dependent on any other factor or any other independent variable or any other factor, that is called an independent variable. It is not affected by any other environmental situation, but it impacts on other variables and dependent variables. It impacts on those variables which are directly depending on this variable. It changes the characteristics of other variables, but independent variable itself is a variable which is manipulative variable which can be changed. There is no impact of any other factor or independent variable. Then comes dependent variable, the dependent or responding. Why it is called responding variable? Because it responds. If we want to change the quantity of sugar, or if we want to manipulate the independent variable, then there is a direct impact on the dependent variable. So whatever changes we are going to make in independent variable, the dependent variable responds accordingly. Now, how is it possible? If we add 50 grams, first we have 25 grams, then we add 50 grams. So every time we're adding sugar, dependent variable, that is the size of the bread, so it is responding, right? Maybe on 25 grams of sugar, its size increases, but on 50 grams, it doesn't increase, or it increased to the max of its capacity. So that is why dependent variable is always a responding variable. It always respond whenever we want to manipulate or change the characteristics of the independent variable. So in this case, it would be the size of the loaf of bread, right? So the size of the loaf of the bread is basically depending on the quantity of sugar here. So that is why it is called dependent variable. Now comes control group. In a scientific experiments, the control is the group that serves as the standard of comparison. So the control group may be a no treatment or an experiment selected group. So basically, what is a control group in any experiment? It is that group with which we compare other groups on which we are doing experiments. This is a group on which all the characteristics of these groups are constant, right? We are not supposed to manipulate them. This is a type of group with which we compare other groups to test the experiments. Either they are accepting the hypothesis or they're rejecting the hypothesis. So this is the ideal group with which we compare the other groups which are under the influence of an experiment, right? So basically, if we see here in this experiment, in this experiment, so what is a control group here? So yeah, if we come to know that, for instance, 50 grams, the maximum loaf we see in the bread is on 50 grams of sugar. When we add 50 grams, there's a maximum rise in the size of the bread. So it means that other 100 grams to 50 grams, 500 grams, while we're adding these quantities, we are comparing these quantities with 50 grams of sugar, right? So that will be the control group. Now, the control group is exposed to the same conditions as the experimental group, except for the variables being tested. All experiments should have a control group. So, whatever the experiment you are going to do, there should be a control group. That is very important for an experiment, right? So, because his grandmother always used 50 grams of sugar in her recipe, John is going to use that amount of his control group in his control group. So, as he always watches his grandmother that she used 50 grams of sugar, so he will change the quantity. But he every time he changed the quantity of sugar, he always compares the results of the experiment with this 50 grams of sugar, right? So this 50 grams of sugar is considered as the control group in his experiment. Now another factor, that is constant. So what is a constant? So constants are those in conditions are those factors which are a constant, which remains the same throughout the experiment. It remains the same for the control group, and it remains the same for the group that is under the influence of experiment. So all other factors stay the same, so that any observed changes in the bread can be attributed to the variation in the amount of sugar. So, what are the possible constants in this experiment? For instance, other ingredients to the bread recipe. For example, if we use oven for 50 grams of sugar to bake the bread. We use oven when we were using 100 grams, when you were using 150 grams. So every time, whenever the experiment was done, same oven and same temperature was used, right? So basically oven was a constant, the temperature of the oven was constant, that remained same for the both of the experiments. Rise time, the temperature he gives for all the experiments. Brand of ingredients, every time he was doing experiments he was using the same brands of ingredients. Cooking time, type of pan he was using, air temperature and humidity where the bread was rising, over temperature, oven temperature, age of the yeast. So every ingredient, every factor he was using while doing experiments were same in all the experiment, except for those he was testing. For example, the quantity of sugar he was testing, so it was not considered as a constant. So, experiment again. John writes out his procedure for his experiment along with his materials list in his journal. He has both of these checked by his teacher, where she checked for any safety concerns. So, trials. Trial refers to replicate groups that are exposed to the same condition in experiment. John is going to test each sugar variable 3 times. So, he's going to, what are trials? Trials are those efforts in an experiment which we are going to do again and again by manipulating the independent variable, the quantity of sugar here in this case. So trials are the number of efforts an experimenter is doing. Either it's 1 time, either it's 2 time or three. So it depends, right? So in this experiment, he was doing experiment by using 3 to 4 different quantities of sugar. So he was basically doing 3 to 4 trials to do this experiment. So collect and analyze data after the careful observations and his proper planning. Then he was able to analyze the results properly. John comes up with the table he can use to record his data. John gets all his materials together and carries out his experiment. So as we have discussed that in the initial slides, that what are scientific methods, the 7 steps of scientific method. So this is the seventh point, that collect and analyze the data, and then interpretation of the results, and then communication of result is a must. So conclusion, John. So what was the conclusion? Sorry, I have skipped this slide. Collect and analyze result. John examines his data and noticed that his control works the best in his experiment, but not significantly better than 100 grams. So he had observed that the maximum amount of the maximum loaf of the bread appears to be at 100 grams of sugar. If he was adding 150 grams, there was no impact on the size of the bread. So the best result he had seen is while adding 100 grams of sugar. So that is his result. So what was his hypothesis? His hypothesis was that adding more sugar into the bread, so as much sugar he was adding, so the size of the bread increases. But in actual, it doesn't happen like that in the way he thought while doing the experiment. The maximum amount of the loaf, or the loaf of the bread, it appears at 100 grams rather than 120 grams, 150 grams. So this is the best This is the best quantity he took for his experiment to see the results. So, conclusion, John rejects his hypothesis but decides to test using sugar amount between 50 grams and 100 grams. Now he have seen that 100 grams is the best quantity for sugar to rise the bread, right? But as his grandmother were using 50 grams as it was his control group, so now he decided to do the retest by using 50 grams, in between 50 grams and 100 grams. So John finds that 70 grams of sugar produces the largest loaf. His hypothesis is accepted. Now, in the second experiment, while he was observing by adding different quantities of sugar, 50 grams, 60 grams, 70, 80, 90, and 100 grams, he had observed that at 70 grams of sugar, the largest loop appears, right? So his hypothesis in this experiment has been accepted.